Good morning, everyone. Welcome to the ground rounds. My name is Geoffrey Hugo. I am assistant professor of medical physics in radiation oncology department. It's my great pleasure to introduce Dr. Geoffrey Hugo from Washington University in St. Louis. Dr. Hugo received his bachelor degree in 1999 from the Pennsylvania State University. He received his master and PhD degrees in biomedical physics from the UCLA. After he graduated, he joined the William Beaumont Hospital and received his ABR board certificate there. He joined the Virginia Commonwealth University, VCU, Department of the Radiation Oncology in 2008 as a tenure-track assistant professor, where he also served as the director of the medical physics graduate program from 2013 to 2017. Dr. Hugo was promoted to the associate professor with tenure in 2014 and a full professor in 2017. Recently, Dr. Hugo moved to the Washington University. Dr. Hugo served on many institutional committee and national society, including AAPM and Astros. He was the president of the Middle Atlantic AAPM chapter and the vice chair of the Therapy Image Committee in AAPM. Dr. Hugo is also a member of the NIH Radiation Therapeutics and Biological Study section since 2015. Dr. Hugo received multiple research grants from the, especially include 2 NIH ROIs as PI. Dr. Hugo supervised more than 10 PhD students and postdoc fellow as a primary mentor, as well as many medical physics students and residents. Dr. Hugo has been invited to give more than 35 national and international talks, has published more than 50 peer-reviewed journal papers and 7 book chapters. Dr. Hugo's research interests include image-guided adaptive radiotherapy, particularly for the lung cancer, image restriction and analysis, as well as machine learning in radiation oncology. Today, Dr. Hugo will give us a talk titled Adaptive Radiotherapy for Lung Cancer. Please join me in welcoming Dr. Hugo.
Thanks, Xiaofeng, for that nice introduction. Can everybody hear me? Is that okay on the microphone? Good. Okay. So just before I get started, I'll my disclosures there. So what I want to talk about today is Xiaofeng said, some of the work that we've done. Mostly this is work at VC, but some work that's moved to WashU and is going to continue on kind of practical adaptive radiotherapy. So what I'll focus on there is kind of tips and tricks that we've learned over the years in studying lung cancer patients to help make adaptive feasible for clinical use. And then I'll talk about maybe less feasible and more kind of physics fun stuff, some of the image registration work we're trying to do, and then some, if we have time, some of the MRI and lung cancer work, which is not the usual application for for MRI, but something we've found some possible some use for. Okay, so to jump in, why adaptive radiotherapy in lung cancer? So I'll show you some of your own slides here to start. Dr. Curran, so we know poor survival in lung cancer. So the take-home message here, rather than looking at like the 2 arms, is just the kind of overall 5-year survival there is pretty poor for locally advanced lung cancer. And why is that? Well, obviously these patients have a lot of distant metastases and things, but we do have a lot of local recurrence after radiotherapy. So just looking at kind of the total percentages here, it's about 50% of patients have a first failure in the primary tumor or in the lymph nodes involved lymph nodes, so in field. So that's a pretty high percentage, and maybe 20 to 30% in field only. So maybe there's some role for improving radiation therapy and improving the or reducing the recurrence rate there. But of course, the question is, with RTOG 0617 results, which showed the high dose, giving higher dose is actually worse, is that the right way to go with dose escalation? I think the take-home message there is to look at, what are people finding analyzing this data and related data sets now. So the question is, why is the high dose worse? Is it due to cardiac toxicity, which we see here is a nice study in like 150 patients or so, where they found that, it was cardiac toxicity definitely had a role in survival for these patients. Or is it something else? Are we irradiating too much of the blood and the immune system, which is a really nice and interesting study that was presented at Astro this year by J.J. Jin. So I think that if you kind of put all these things together, the point is, we need to really consider normal tissue toxicity, which is not anything that I'm not telling you anything you don't know already. But if we want to do something fancy, like dose escalation or any kind of functional targeting by using PET or something to identify a radioresistant area, or looking at immunotherapy and targeted agents where there may be additional toxicity when we add radiation to that. So there I think the issue is, how do we reduce the normal tissue toxicity beyond what we have today so that we could do these fancy therapies? So that's what I'm going to concentrate on today, is how might we try to get higher precision and reduce the normal tissue toxicity. So our thoughts are, if we look at these patients over time, so this is kind of just a little movie loop of 5 or 6 weeks of treatment with a single plan that's not been adapted. And so you can see the tumor here in red, and I threw some of the normal tissues, so the esophagus in yellow and the spinal cord in green. You can see those are moving around, changing a little bit, but the real key piece that you see is this tumor is shrinking, probably in response to therapy, but we're still irradiating, a very large field, and most of that's is probably not tumor in here. It's probably lung or some, who knows. So the question is, how can we reduce the field? How can we be more precise and track what's really going on with the anatomy? If we have higher precision, this should lead to less toxicity. This is our hypothesis. And we could also then get better estimates of what we actually delivered. So if we're tracking dose over time, tracking where the anatomy goes, and using that to adapt, this will allow us to better understand what dose we deliver to the patient, and we could feed this into outcomes models, for example. Though that's not the primary goal of what I'll talk about today. Okay, so how are we going to do that? How are we going to get higher precision just by looking at how the anatomy changes? The concept here is called adaptive radiotherapy. So the basic idea is, we start with imaging, we do planning, we treat the patient. 6 months or so later, we get an image, a CT scan or a PET scan, see how we did. So the concept here is not anything we developed. It's been around for a while, but the basic idea is now, while you're treating the patient, you're getting imaging, you're performing assessment, so you're seeing what's changing, and then you're feeding that information back into the planning process in a feedback loop like this. So this is the basic idea of adaptive. To do this more in not really real time, but while we're treating the patient. Okay, so adaptive's been around a long time. I think around 20 years ago was the first paper that talked about adaptive radiotherapy. So, because it's been around a long time, is there any benefit? May this actually help us reduce toxicity or improve precision and improve outcomes? So this is a really nice study from a group in Denmark, where they actually did a clinical trial to compare adaptive radiotherapy to no adaptive radiotherapy in lung cancer patients, and they found a significant decrease in failure rates in the adaptive patients, which you can see over here. So this is done. Why are we going to have an hour talk about it? What we need to do with adaptive in physics? Well, this is a very manual process. So they basically had a group of therapists go through and review cone beam CTs every day, along with physicians and physicists making decisions about what to do for that given patient. Then they had to go through a highly manual replanning process, which is going to be very cumbersome and time-consuming to do. They did find that they need to replan about 27% of their patients once, and a small proportion more than once to achieve these results. So even with 1 or 2 replans, it may be that this is not going to be feasible for your routine mom-and-pop shop to do. How are we going to make this process more efficient, number one? And number two, is this optimal, this manual process? Are they really finding all the patients that need to be adapted? So this is what I'll talk about with our work, is this concept of replanning the patient on the fly is still quite costly, and it's quite complicated. There's safety issues involved anytime we have to redo a plan and run through all of that process. So we want to make sure this is going to be safe and effective. The other question is, when do we adapt? So how do we analyze the data? How do we support decision making for the physicians? If a physician has to look at a bunch of cone beam CTs and make decisions about planning, is that really the best use of time, or can we develop algorithms to help with decision support? So I'm not going to show you that level today, but kind of getting to that point, how are we going to do that? And then how often to adapt? So should we just schedule every patient be adapted every week? Should we analyze images, as I said, and try to characterize how things are going? Or is there some other way to do this? So trying to assess that and answer that question. And then safety. So every time I give a talk about adaptive, I get a question about, aren't you concerned about shrinking the fields too small and having a miss? And so this is a real issue to be concerned with that we have to combat. So how do we do this safely and make sure that we're not overdoing it, basically? Okay, so we really wanted to focus on the patients we think are going to have most benefit for adaptive. So if we looked at like early stage patients that are getting 5 fractions or 3 fractions of SBRT, there's probably not going to be a lot of changes. So the locally advanced patients and a patient that has very clear lungs and the tumor is very, it's a very well circumscribed tumor. You can see it, it's not changing very much. The patient's breathing normal. Those are probably not the patients that are going to have the most benefit because they're not going to have a lot of changes. So as I'll show you in a minute. So we think kind of the biggest issue is to look at these kind of patients, the patients that have atelectasis, which is partially collapsed lung, often adjacent to the tumor. So this is kind of a typical case here. You can see here's a tumor which has been identified with PET. And this is the first week or the planning CT. And you can see this big area of collapsed lung adjacent to the tumor. And as we treat this tumor, it starts to shrink. Airway reopens, and we get re-expansion of all this collapsed lung, as you can see here. So big kind of geometrical changes to the patient's anatomy. The question is, how much dosimetric effect does this have? So does it change the dose delivered to these structures? Are we estimating the dose correctly at the planning CT or not? So what we found was just looking geometrically that these patients that present, at the time of simulation with these big areas of collapsed lung and things like this, they often have large changes during treatment. So we presented some work at Astro in the past showing that patients that had atelectasis at the time of simulation versus those that didn't had much larger tumor shifts, something like 5 millimeter shifts in 83% of the patients or more, and large volumes of changes of atelectasis. And as I'll show you on the next slide, it's associated with sometimes large changes in dose. So what you're looking at here is 2 patients, one patient in the top row and one patient on the bottom row. And so this patient on the top row, you can see the target, the tumor, primary tumor in red, and one of the lobes that had some collapsed or atelectasis in blue. And during treatment, you can see that the tumor had shrunk after some portion of time, and this lobe had re-expanded. So this is all manually defined by a radiation oncologist, these contours. And then in the bottom, a patient that maybe had some changes in atelectasis here, but it didn't really induce very large changes in tumor position or in the lung dose. But the take-home was overall, we looked at 20 patients, and you can see 2 different alignment methods. So if you're simulating aligning on bone or simulating aligning on, say, primary tumor and carina, we did see a number of patients that had somewhat large changes. The averages are not huge, but there's a subset of patients where we can get pretty large changes in the dose to the cord, the esophagus, and the lungs. Okay, so I think these patients are clearly patients that are going to have, we want to watch because there's going to be changes and it could have a dosimetric effect. But there's another consideration to take, another consideration here, and we're often treating with locally advanced patients involve lymph nodes in addition to primary tumor. So the primary tumors tend to shrink over treatment, as you can see here, but also the nodes tend to shrink over treatment. And we showed, I'm not going to show you the results here, but roughly the change in volume by percentage is pretty similar for primary tumor and lymph nodes. It can be something like they can lose almost half their volume over time, but it varies wildly patient to patient. But the implications of this are not only these volumes changing, but we can get some shifts between the two sets of targets. And if we're treating this all in one PTV, it's kind of deforming our target structure then, so how do we account for that? What we found was, if we look at the distance here between, say, the primary tumor and the lymph nodes, so consider the distance between those two, and looking at how that changes over the course of therapy by re-imaging these patients with CT, we found that patients that had no atelectasis, the average shift and the range of shifts here day to day was around 2 millimeters. So they don't really change very much. There's not a lot of shifting between the primary tumor and the lymph nodes, even though breathing patterns can change and things like that. Whereas patients with atelectasis at the beginning of treatment had more like a 6 millimeter on average shift, and you can see these vary quite a bit. So this is probably just due to the fact that the atelectasis is changing. It's pushing the primary tumor around in the lung, whereas the mediastinal structures are staying anchored, and so you see much bigger shifts here than in this group of patients. And we said, okay, well, what happened if you could find the time where maybe that atelectasis reopened and you could replan at that point in time? So just assuming that you knew, beforehand when that was going to occur. Of course, that's not realistic. But if you made kind of that optimal single correction, you could reduce this variation down in these atelectatic patients to about 2 to 3 millimeters as well. So this is kind of giving us some evidence that maybe there's a time point where if we could really see where the changes are occurring the greatest, with only 1 or 2 adaptations, maybe we could make some real benefit here, at least geometrically. So this is a little more sophisticated study along those lines. What we did is we had weekly CT scans, 40 CT scans in this cohort of 20 patients. And so we using some modeling techniques, we went and in image registration, we produced synthetic 33 fraction courses of imaging in these patients. So we basically built a model with these weekly images, and then we're able to create a bunch of different simulated treatment courses geometrically in these patients. So what we did then is we said, if we adapted every single day, okay, nobody'd ever do that in the clinic, but if you did, and then you delivered it perfectly, so we had basically, this 33 fractions of imaging, and we made a plan for each individual image, and assumed that we delivered it perfectly, that's the most precise we could ever be. It's not realistic, but it's the best we could ever do. So if we, using that, looking at what the normal tissue dose, how far we could reduce it, or how far we could increase the dose to the target with that approach, that was our kind of maximum benefit, we'll say. The most we could spread those two quantities. And then we looked at, okay, now if we only did 1 adaptation or no adaptations, or 2 or 5 or so, how much of that total benefit could we recoup? And so that's what you're seeing here. So adaptive frequency is basically the number of times we replanned, up to 33 here. And then on this axis, you see the percentage of this full benefit. So how much of that, let's say if we're escalating dose to the target as high as we can go with the daily adaptation, how much of that benefit dosimetrically can we capture? And so what we found was about 2 to 5 times of replanning. So that'd be 2 times during the course. 5 would be roughly weekly for these patients. We got about 90%, somewhere between 70 to 90% of the benefit. So even not knowing anything about when that large change occurred in this group of patients, we could get quite a bit of the benefit. So maybe we don't need to do daily online fancy adaptation. Maybe we can just send the patient back for simulation periodically when we observe something like a change in atelectasis, or just have a fixed frequency already scheduled a couple times during therapy, and we may get quite a bit of this benefit. Another group did a similar type of study, but what they were looking at is when is the optimal time to adapt. So if you're going to do a fixed schedule like this and maybe adapt once or twice, when should you do it? And they found that after about 3 weeks of therapy is the best time to get the most bang for your buck. Okay, so as I said earlier, we have a concern when we're doing these type of study, or this type of approach of shrinking too much, like making the fields too small. So how do we counteract that? Because if we see the primary tumor shrinking, we'd really like to reduce our high dose area to that region. just treat what we see either on anatomical imaging or functional imaging as viable disease, right? But there is the concern that, as we're shrinking, we leave some microscopic disease in the wake of this area. So how do we counter that? So we've got this concept that we've looked at to look at the CTV. So the basic idea is, when we treat these tumors, these large tumors, the primary tumor in red, we expand by some fixed margin to account for any microscopic extension into the normal tissue. And so you can see that subclinical target, or the CTV there in green. So, this is presumably the maximum extent. I know it's not perfect. It's an estimate for this patient, but presumably this is what we'd really like to sterilize, all of the tumor cells within this green region. So the question is, as this tumor shrinks, where did this green region go? Is it adjacent to the tumor? Is it still out here somewhere? So the nice thing is, at least where this extends out into the lung parenchyma, there's lots of vessels and airways and things that we can see that are unchanged on anatomical imaging from day to day. So using image registration, we can track where these structures go, and then knowing kind of the relationship between those structures and this subclinical target here, we can map this onto this later image. So the yellow is basically this green structure that's been mapped onto the later image by image registration. And you can see that, in some regions it's still about the same distance from the tumor, and in some regions it's quite a bit farther away. So the question is, what's going on here? I don't have a good answer for that. I think some of it's just a geometric issue, that if you have a concentric spheres and you shrink the inside one, the outside one can't geometrically shrink as much. And then some people think that maybe these tumors are disintegrating away. I don't really think that's probably what's happening, but we don't really know. But that's a little bit more of an interesting scientific question. The practical question, or the practical issue, is if we know the green is what we want to treat to a sterilizing dose, if we can still give that dose to this yellow region down here, and then boost this red region as high as we could go, because we presume that's where most of the viable disease would be, that could be a strategy for counteracting this issue of shrinking the target. And so this is the concept that we are developing into a trial. So basically, here you see the PTV in red for your primary tumor and your lymph nodes. And so we'd go up to, say, 50 gray here, and then we'd adapt, and we'd map this in green, this deformed CTV that I just showed you. So we'd have that mapped, and then we have our residual viable disease somewhere inside this, what we see is still tumor on the CT image. And so in that blue region, we'd boost that as high as we could, and just keep this red target out here to like a standard 66 gray dose. So the idea is we kind of have a field in field, but we try to treat still that original field to the 66 gray and just boost the GTV. So I don't think I'm going to show this, but we did a planning study in 10 patients to show that if we use some like ARC's VMAT, for example, comparing 66 gray, like a standard dose, to this red target, which would just be a conventional therapy, to doing this approach with the MAP CTV and then boosting this blue area here, we actually could do that without increasing the dose of the normal tissue very much. It was pretty minimal increase in lung dose, and no increase to structures like the esophagus and spinal cord. Just because you're basically, when you're delivering a uniform dose there, it's actually somewhat harder sometimes than allowing the system to just give a much higher dose in the center to this region here. So it didn't, kind of maybe contrary to common sense, it actually is not going to increase your normal tissue dose much to do that. Okay, so this is a strategy that we think is a potential way to do adaptive safely, maybe alleviate some concerns. So we're going to try this in a trial soon is the hope. So for this practical part, I think the take-home messages are, if nothing else that you take home today, pay particular attention to these patients with atelectasis. They tend to have larger changes, both geometrically and dosimetrically, and are probably the ones that are most likely to benefit from adaptive. So they're probably the patients to concentrate our efforts on the most. Maybe only a handful of adaptations is all we need to get most of the benefit. And so, instead of trying to build tools where we can take a cone beam and redo the plan every single day online, maybe it would be a better use of our time to try to make this offline process where we get our simulation and things more viable, and only shoot for a handful of high-quality adaptations. And then, of course, we've got the issue of the primary tumor lymph nodes and the original CTV, as I went over. Okay, so now I want to change gears a little bit and talk about, well, how are we going to make this work in the clinic? I showed you that nice trial from Denmark where they're getting good clinical results, but is this really feasible to do on a day-to-day basis in most clinics? So we would say we probably need some tools, as most people think, like image registration to help this work. How are we going to make image registration work for these really tough cases with atelectasis? So the first thing is just to talk a little bit about if you don't want to do all the fancy image registration, how would you start? So at VCU, we took this concept of a traffic light protocol that was developed at NKI. So basically, we do a kind of semi-subjective, subjective with a little bit of objective rules analysis of cone beam. So this is all manual, but basically the idea is the therapists have a set of rules where they would review the cone beam CT here. And you probably can't see it very well, but in pink there's a PTV and in red there's a GTV. And so basically green light means that the image looks the same on cone beam as planning, and so they can proceed to set the patient up with image guidance and proceed with treatment. And then the yellow light means there's been a change inside the PTV, but there's no obvious areas outside the PTV that have changed. So yellow would be go ahead and treat, but let the physician know. Orange is there's a change outside the PTV here. And so that needs to be, so orange and red basically means some large change outside the PTV. We consolidated this down at VCU into 3, just a green, yellow, and red. But this would be, contact the physician and don't proceed until they've given a decision about what to do. Oftentimes this would be a patient that we'd send back for resimulation. Okay, so this is a nice kind of semi-objective way to do things and to get it piloted in the clinic. And we're doing this, starting this up at WashU now because I think people have found it to be pretty valuable. But the question is, looking at these cone beams, it can be really difficult to see, where the anatomy really is. So I mean, it's hard to kind of here. Maybe these two, it's pretty clear, but up here, what's going on here? You can't really see anything that would tell you where exactly the target is over here, and clearly up here, there's a lot going on. So can we really make a decision about this just from this cone beam? Or should we use image registration to help us and then send this patient back for resimulation and use that data for planning? I think that's the question. But the ultimate idea is we hope that we can develop image registration tools that would be able to automatically analyze these images and help us with decision making about what to do, contouring, and then summing the doses together from day to day to get a better picture of delivered dose. So the challenge there is that traditional registration algorithms aren't really built to handle this kind of problem where we've got, say, the tumor here and maybe some atelectasis or something else here, and then mid-treatment, not only has maybe this region resolved and then we've got something new up here, or maybe it's shifted, but this really challenges a traditional algorithm. These algorithms are generally built on the concept that you can take an object in an image and kind of squish it and pull it and make it into the object in the other image. So we call this topology change. So if you can do that, if you can kind of just push and pull and stretch things around and make the image look the same, that means that it's not been a topology change. Whereas here, if a piece of tissue has really changed appearance, if a piece of tissue's disappeared, like the patient had a surgery and there was some tissue resected, that's obviously there's no way to push and pull that image and make it look like the other image, right? So under those kind of topological changes, it doesn't meet the model of how these traditional registration algorithms have been built. Okay, so we need to maybe build new tools or modify them in a way to make them fit this model or fit a model of what's actually happening here in these images. So our idea, our solution to this is to kind of try to, without making some fancy new math of how to deal with this, is very practically just to can we ignore regions? Can we identify the regions that have these topological changes or appearance changes in the image? And if so, can we kind of ignore them in the registration? So either find the regions that are not consistent and ignore them, or find the regions that are consistent, things like vessels and airways that we know are there in both images and really aren't going to disappear just by airway or lung re-expansion or collapse. And if we can identify those consistent regions, then use that to drive the registration and then build some models or interpolate what's going on in the other regions. So to put this kind of concretely, what we're doing is trying to segment vessels, airways, and then lung lobes. So we know those are all there. They may just be hidden in the image by this collapsed tissue, and register these regions, and then model the changes in the other regions in, say, the tumor, although that's getting ahead a little bit, but in the atelectasis and if the patient has a fusion or something like that. So try to model what's going on there. And I'll show you, actually, maybe we don't even need to model, because if we have enough consistent anatomy around there, it actually does a good job getting the registration right, where we care about getting it right, and the models don't seem to help all that much. Okay, so how are we going to extract vessels and register them? There's a huge body of literature on how to highlight vascular structures in medical images. There's this really cool paper in Medical Image Analysis. It's actually a living review. So even if you don't care about vessel segmentation, it's neat to see what they did, because, you know, if you read a review paper, the minute it's published, it's kind of outdated, right? Because people are still working on things. So this concept is they built this website that you can continually add papers to this review, and you can sort things by different, for example, like, was it a vascular segmentation on a CT or an MR, or, you know, what's the application? So just a cool concept in general. So I'd say check out this paper, even if you don't care about the vascular image registration. But there's a lot of ways to do this, and in lung there's a lot of algorithms that have been built, so rather than reinvent the wheel, just use the best ones we can find. So basically there's two kind of ways to do this. You can treat the vessels as an image, so try to enhance the vessels on an image, and then still you have an image here where you can see most of the anatomy is gone, except you can see the bright white where the vessels are. It's really just a filter that runs on an image and makes it enhances vessels, or you can treat it as, I'll call it a tree. So you actually segment the vessels out, and you have segmented structures. So you no longer have an image now, you've got like a 3D model of the vessels. So two different ways to do it. We've looked at both methods. I'll show you some results from both. So this is what I just said. You know, you can extract it out with a filter approach. So this is an example of doing this approach with the image-based approach, where we've just filtered out the vessels and still registered these. So once you do this, the nice thing is you enhance the vessels. You can give this to a traditional algorithm like a MIM or a Velocity or one of these open source tools, and just let it register these images instead of the original images. So it's a simple way to get started. We did some work with collaborators at University of Iowa and found this works pretty well for like small structures out here, where this is just an original registration, and this is the original image. When I say original, I mean image-based, like just registering the two images as we normally would, versus doing that and doing including this vessel-based registration as well. You can see what it did is it basically found these little areas out here where there's small, not a lot of contrast in the original image, and it kind of repaired that and got a better registration. But, you know, not really groundbreaking. It gives you a little bit better results, but this wasn't really our use case. This is more just end exhale and inhale type registration, so not really with big changes. This is more looking at our application. So here we extracted the vessels, we segment them, so we've got these 3D models, and then we can use some basically fuzzy matching approaches. So instead of trying to find exactly correspondence between the vessels, it kind of does a probabilistic approach without getting into too much of the engineering of this. If you're interested, I can show, you know, the physicists want to see equations and things. I can show you some of that later. But the basic idea here is what this algorithm does is called varifolds. It tries to, without doing one-to-one. It's trying to match up areas that look similar, but it's capable of ignoring if you have mismatches. So where this would occur is, say, part of the vessels, like here I have a intact vessel tree in my hand, and in the other image, there's some collapsed lungs, so some of these vessels are obscured. And so this algorithm I should probably do it this way, but this algorithm is capable of kind of lining this up and ignoring where there's not correspondence. And so that's why we use this algorithm for this approach. And so what you can see here is an example. These are the before and after. So what you can clearly see is like this green lobe here is partially collapsed here, and it's re-expanded here. So there's a lot of missing vessels in this image that are present over here in the after treatment image. And then this is the looking at the deformation, and it may be better to look at down here. You can see before registration there's a really big difference between the vessels in these two lobes, and then after registration it's not perfect, but we get it aligned a lot better. And when we try to use a traditional algorithm for this, it's not capable of doing this because there's just too much change here and too much mismatch where there's missing vessels in one image and not in the other. Okay, so what about inside the atelectasis? I said we need a model there. So what's the model we're going to use to expand atelectatic tissue and make it match? So our hypothesis is, okay, if lung tissue is collapsed down in, on an image and it looks, nice and uniform and high density, well, when it re-expands, it should still have the same mass, right? It's the same tissue, you just have collapsed it down in the image and then re-expanded it. So if we can use what's called a mass-preserving algorithm, there's algorithms that actually implicitly take into account the preservation of mass and therefore the density changes. So if you expand tissue, as I'll show you here, this is a sheep lung, but you can see when it's collapsed down, the intensity and therefore the density is much higher of the tissue, and as it expands, right, the density decreases. So in this case, mass is generally preserved. I mean, there's some blood coming in and out of the lung that maybe changes that somewhat, and of course it's quite heterogeneous across the lung. But in general, over the whole lobe, it should be preserved. So we think this is a reasonable approach to apply to our problem. So the first question was, well, is mass actually preserved in atelectasis? So the best way that we could answer this question was to try to delineate the lobes and then look at the change in mass between, say, pre-treatment and a mid-treatment where we've got a clear change in atelectasis on a patient. So is the lobe mass preserved? And what we found was we could, each of these patients has like a contralateral lung that generally is pretty clean and doesn't have atelectasis and tumor, so we could use that as a control. In the healthy, or in the ipsilateral lobes where we're trying to correct for this, we had to try to get the tumor, segment the tumor out. So it's a little challenging, there's some error there, but if you look in general, we see that in these control lobes, contralateral and healthy ipsilateral lobes, there's not any change in mass, even though these, there may be changes in volume. And when the lobe is fully collapsed, so the whole lobe's full of tumor and atelectasis and re-expands, we see that on average, although there's quite a variability across subjects, there wasn't much of a mass change. Where we do see some actual mass loss, so basically as this lobe re-expands, we measured that there was a loss in mass in some of these patients that had partial lobal collapse and then re-expanded. The best we can estimate what's going on there, and maybe a working hypothesis, is these patients, it's not just collapsed lung. That's a really simple physics assumption. And these patients have infiltrate and things like this, and so as the lung re-expands, and then you wait a little bit of time, like some of this is gonna resolve, and so all of that gunk that's in there is gone. So that's what we think is happening there. But if we look at intensity change, we have huge intensity changes, as you've all seen on a CT, and I just showed you. When the lungs collapsed, it's a very high intensity, and as it reopens, you see the low density parenchyma as well as the high intensity vasculature. And so still we think using this mass preserving cost may work better. As I'll show you, it actually doesn't, but that was the thinking when we started this project. So put it all together, what are we actually going to do? We've got a multi-resolution B-spline framework. We use the open source registration algorithm because we can plug our components in as we develop them, which is really nice. So anywhere where there's healthy lung, it should work the same as that sheep lung. And so we use this mass-preserving term there. So there in blue is the healthy lobes, or the, let's say, non-tumor bearing and non-atelectatic lobes. So healthy parenchyma gets mass-preserving cost. In the atelectasis, in those lobes, as you can see here, we're going to try to do a couple different things and just ignore the acronyms. Basically, the first two are mass-preserving, the bottom two are conventional. So, does the mass-preserving term work or not is what we're trying to test with this approach. And then we're going to use lobe labels. So we can delineate the lobes themselves. So a physician, for example, can go and delineate this. We can use some semi-automatic methods. This helps kind of get things initially close, because these algorithms, or these changes can be so large that they can confound any algorithm that we really throw at the problem right now. So we start with this and test this as well, and then the vasculature, which you can't really see down there, but there's our segmented vessel trees. Okay, so we're going to throw all this together, and then we're going to test plus and minus, meaning which of these components are important and which aren't. And looking at patients where we didn't have a big change in the atelectasis, a partial change, and then, as you can see, a full change here where there's a big change in the atelectasis. So we saw similar results, errors of about 2 millimeters there. And so this is looking at, in a cohort of 18 subjects, 18 image pairs in different subjects, all these different plus and minus, these different components. So this was using basically no lobes and just looking at the different metrics in the atelectatic lobe, so the different models basically. And then once we add lobe labels in, you can see we reduce the landmark error. So this is a measure of accuracy, so lower is better. And so you can see we're much better accuracy, much higher accuracy when we add the lobe labels. Adding the vesselness term to the lobe labels reduced some of the outliers a little bit, and so we think that's beneficial. But the big take home here was you have to really get the problem started right by using the lobe labels to get things close, or there's not much hope in getting this right for all patients. And then, so what's the error kind of close to the tumor and atelectasis versus farther away? So this region means the smaller this is, the closer we are to the atelectasis and tumor. And as we go farther away, we didn't see a big change, actually maybe a little bit of an increase in the error, so decrease in the accuracy as we go farther away. And the contralateral lungs are doing a little better, as we might expect, than in the ipsilateral lungs, but not too much decrease in the mean across there. Okay, so this is a little busy, but it's basically, what's the take home? Use the lobe labels. It starts the problem off right. It gets us close at least. So we found that to be a big benefit. So unfortunately, that's a lot of manual work, but if we think about we're only going to do this maybe once or twice during treatment. And so if we have a new planning CT, we ask someone to delineate the lobes, and then we can use an automated algorithm to kind of help get everything else right. That would be probably acceptable for us in the clinic, at least to start. Unfortunately, we didn't really see that there was any need to do fancy modeling inside the atelectasis. Getting the lobe labels right, adding the vesselness, and then getting the registration right in the healthy lungs seemed to do just as well no matter what we did with making a model in here. So, the good news is we can probably throw that away. We don't need to build a model of how the atelectasis expands and collapses. Okay, so I think, at least at first glance here with this study, doing some lobe segmentation, adding in some of these fancy cost metrics, but maybe not modeling how the atelectasis expands and contracts, gives a reasonable error of about 2 millimeters, which is what people see for intact lung when they do day-to-day registrations without these large changes. But we're going to certainly further develop this to try to automate some of the segmentation or improve the algorithm so we wouldn't need to segment the lobes individually and include this in the registration. All right, so maybe in the last 5 minutes, I'll go through a couple of things we've done with MRI, because I know you guys do a lot of work with quantitative imaging, and we're interested in this area, particularly for lung cancer, so I'll just work through this really quick. So I've shown all anatomical guidance right now, and we'd really like to hit the residual GTV with, the viable tissue should be in there. But the question is, most of that's probably not viable tissue. What we see after a few weeks of therapy as the residual GTV is probably either dead, mostly dead or dying tissue, and there's pockets of viable disease. So there's already, a lot of work going on with using, say, FDG PET and clinical trials with NRG that are looking at trying to hit this residual area. We think this is a great idea. The functional, looking at what's actually functional inside, what's actually viable would be a good idea, and maybe we could hopefully then not have to increase the dose to that whole residual or tumor, but maybe pockets of it, which would make it a little bit easier and maybe less normal tissue dose. But we've looked at things other than PET. PET does have specificity issues. A lot of times if you look at a mid-treatment PET, there's inflammatory effects around the tumor. The tumor PET signal after treatment or in mid-treatment can be so low it can be hard to distinguish the two. And of course we know there's resolution issues with PET, especially with breathing motion and things. So we've looked at MRI for this, particularly or specifically diffusion-weighted MRI. So this is not a new concept to use diffusion-weighted MRI as a measure of residual radioresistance. So this hypothesis from Brian Ross is basically like as you start therapy here and you've got a highly dense, highly cellular tumor, and so if you measured the diffusion of water in that tumor, it should be fairly low because there's a lot of structure and stuff here, and the water can't move or diffuse as far. As you start to treat and as this tumor starts to break up, you get, and especially in areas of necrosis, the diffusion should be higher, and so you should see a higher signal in diffusion-weighted MRI. And so people have tested this in all kinds of different sites, cervix and brain and things. So in lung, we thought, well, why not look at lung and see if it works there. There was a good bit of technical work to get a sequence that could actually perform with respiration. So we looked at both breath hold and respiratory triggered. We didn't see much difference between the two. It's easier for these patients to do respiratory triggered free breathing. So that's what we proceeded with. And I can show you a little bit of the repeatability data in a second. But basically, in this subject, you can see here is the tumor in red on diffusion-weighted MRI pretreatment. There's little pockets of high intensity, but for the most part, it's fairly hypointense. And then as we start to treat, you can see that the average intensity inside the tumor gets much higher, as well as it's shrinking. So the red is the original target, and the pink is the GTV that we see after 50 gray. And so you can see it's increasing on average as the hypothesis showed. But this is one patient. So I'll show you in a minute a handful of patients that we've imaged. But repeatability is really crucial with this. So if we can't get a repeat the sequence in the same patient, even on the same scanner, then there's not much hope of being able to do this in multiple clinics with different scanners and sequences. So we measured, with coffee break, basically this means get the patient off the scanner and put them back on and see if we get the same measurement. We're unlikely to see a real change in signal over the course of a few minutes, so it should tell us how repeatable things are. So in 7 subjects, we had 25 repeat scans. And this does include things like different respiration. We tried, breath hold and respiratory triggered, as I said, but also different slice thicknesses or slab thicknesses and things like this to try to see if this is repeatable. So that includes kind of all this together. And so in kind of normal tissues that we expect to be pretty stable, we get a 3% or so variation. So if we think about, a true measurement, maybe above twice this baseline. So if we saw something that was, say, 6% or so, we could say, well, that might be a true signal. And in tumor, it was a little bit higher, but these tumors there's a lot going on, as you saw here in this patient here. There's quite a bit of variation inside the tumor, so it depends, if we made the measurement exactly the same, but a little bit higher repeatability there. But I think, it's feasible to proceed. So this is looking at, well, how does the does this signal, this ADC signal, actually change in patients? So really preliminary data at this point, just to show you kind of what we're seeing. And so this down here is the percentage change in ADC over 3 weeks of measurements for these patients undergoing, let's see how many patients do I have there, 10 patients, I think, patients undergoing radiotherapy. Of note, these two that are red dotted lines were patients that were considered non-responders to therapy. Everybody else was a partial or complete responder. So, no statistics on this yet. It's early. We need to collect more data. But it does show some promise that, maybe some of the patients, we see at least a different signal. So it's feasible to measure ADC change in lung cancer, in lung tumors. So I think in the interest of time, I'll skip through and just this last slide. So, beyond like looking at assessment, MRI might be really useful in lung for delineation. These tumors, we often use PET or pretty much always use PET to help delineate the primary tumor because it's often attached to the mediastinum or the hilum or there's atelectasis adjacent to it. It can make it challenging to delineate. And so looking at some of these MRI sequences like a VIBE, which is a T1 weighted scan, you can clearly see a lot of the tissue boundaries. So the question is, could you use this for delineation to maybe to help with PET CT delineation or replace it? So the first question is, how well do we do just kind of asking radiation oncologists to delineate these? The answer from this study was not all that well. So if we compare like a CT only, which of course isn't really used, to a PET CT based delineation and looking at inter-observer variability in the delineation, you can see here all these different contours from different radiation oncologists. And PET CT was clearly the best, but everybody's used to doing that. Everybody's been trained to do that now. And so when we throw MRI at them, even when we have radiologists try to help them kind of identify and do some training, they didn't do quite as well. They did better than in CT only, which was good. But I think the take-home message here is, if we want to try to do this, the images look great to a physicist, and we think maybe they'd be useful, but we probably need to have a training protocol and really get some training on how to look at MRI in the thorax and how to delineate. Okay, so I think I'll wrap it up there. I think, clearly we need to watch these patients with atelectasis, look at how we're going to do adaptive in those patients, try to improve our tools there. They're most likely to benefit from ART, but as you saw, they're the hardest to both register and then probably to replan once we get to that point. So, maybe we're finding these tools for those patients would be useful, and everybody else we can use conventional commercial tools. And I think, incorporating things like PET, which people are already doing, but MRI and all of these into this, fold this into the adaptive process. So, one of the challenges I think now is that people are doing PET guidance, but maybe not doing as sophisticated of geometric adaptive, but combining these together is probably the way of the future. And so collaborators at VCU and also WashU and as I said, at Iowa and a few other places, they did all the work, so I'd like to thank them quite a bit. Thank you very much for your attention, and I'd be happy to answer any questions if you have any.
Very nice talk. In your slides simulation, you showed one figure for the adaptive planning benefit. So what kind of image modality did they use in that study?
Oh, okay. Yeah, that was all CT based. So I can talk a little bit. Let's see. This one here. Yeah. So this study was we had repeat 40 CTs, and we performed image registration week to week to get kind of a trajectory of the tissue. And then we built a principal components model of that so we could statistically resample that data and make different treatment courses. So this is not just 20 subjects, one course. We actually had, I can't remember how many he did. It might have been 10 to 20 simulated courses for each patient to get some statistics. So yeah, I don't have any error bars on here, unfortunately. I need to put them on. But so there is variation between patients and amongst a single patient as well.
You see what kind of the image modality will give maximum?
I don't think there's one. That's kind of what I was getting at the end. I think probably CT in lung is really good at measuring the changes in the anatomy because we can get it daily. And MR, like what I've seen for MR-guided therapy so far in lung, it's maybe not quite there. I think we'd still prefer to do a 4D CT on a CT simulator. But I think combining everything together in some way would be great. But it's questionable whether that's feasible. Like, are you going to send a patient back at week 3 to get a simulation CT is fairly straightforward to reschedule them, but are you going to have them do another PET and another MR? I don't know if that's entirely feasible outside of a clinical trial. So I think that'd be a good question to answer. Is, what imaging do we need and at what time point do we need it? So.
Let me ask a question.
Sure.
So when you segment the vessel from CT image, did you use 4D CT or 3D CT?
So some of those, I didn't mention that. Some of those patients in that study are 40. We took whatever we had, basically. So maybe roughly half those patients are 4D CT, and the other half are breath hold. So we had a protocol a number of years ago where we scanned repeat breath hold CTs with ABC, with the active breathing coordinator, to try to characterize how variable it was in lung patients. So we had that data on hand, so we used that for the study. I don't think we didn't really quantitatively compare it, but we didn't really see a better or worse performance in either. I mean, the breath hold scans don't have any artifact, and some of the 4D CT scans do. But we selected out a single phase, I think, end of inhale to do for that particular study. Yeah. Yeah, that's a good question. So, I mean, a lot of this was done before. That was like the hot topic. But we have gone back and done an analysis of like the variation of the heart over these 4D CT scans. It's challenging because in a 4D CT, it's not really geared to image the heart well, and so you get a lot of blurring artifact and sorting artifact around the heart. But we figure if we delineated the heart on all 10 phases of a 4D CT, we could get kind of an average picture of what the heart looked like. So we have done that. I can't recall like what the variation was, but we did see some, a couple gray of variation in the mean heart dose, for example, over treatment. But that's the level of what we've done so far, is look at variations. Yeah. I think that's an interesting area because people are very interested in those couple studies where we look at what the toxicity might be in relation to the dose. But the question is, how does it vary over time? And if you had a better estimate of the delivered dose to the heart, would it change things, number one? The other thing we've done is we looked a little bit at like what their, probably not the right word, but like the cardiac status beforehand. So if they had cardiac events before therapy and then after therapy, and so there's quite a variability in those patients, and it seemed like there was some correlation, but we didn't have a lot of data, so.